Okay, students, welcome back to part 3 of the cellular respiration lecture. And the last thing we're going to learn about in the process of cellular respiration is the third stage, which is called oxidative phosphorylation. So, in the previous lecture, right at the end, I mentioned that oxidative phosphorylation occurs across the membrane right here. And that is the inner membrane of the mitochondria. Remember, that's a phospholipid bilayer. And that phospholipid bilayer represents a barrier to set up a concentration gradient. So we're really going to be concerned with what's happening here in this intermembrane space and here within that inner membrane. Those electrons of the electron transport chain are going to be embedded in this membrane right here. And so essentially electrons are going to get passed from one protein to the next to the next. And remember, when electrons are passed down that electron transport chain, energy is released. And it's that energy that's going to be used to create ATP. So back. So here is that intermembrane space. Here's the membrane. And then here is the space within the cristae. So we're going to essentially, or in this picture, we've essentially zoomed in on this area right here. We're going to move to this picture, and this picture is kind of scary at first, but if we just take a moment to understand what we're looking at before understanding what's going on, it will make a whole lot more sense. So I'm not sure why the authors didn't do this, but out here is the intermembrane space. Here's the inner membrane. I'm just going to abbreviate that IM, and this would be within the cristae, within those folds. So let's have a look. Remember that all membranes are made of phospholipid bilayer, and so you can clearly see those phospholipids here, right? Another thing you see are the proteins that act as the electron carriers. They're here in purple. So there's one here, 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 here. All of these purple proteins are electron carriers. Also in the membrane, embedded in the membrane, is another protein, an enzyme, actually, and all enzymes are proteins. That's called ATP synthase, and that's going to come into be important at the very end of this process. Okay, so let's go back to the process and how this occurs. Let me go ahead and erase a bunch of this stuff that just makes the picture busier. We don't need that. It's already busy enough, right? So let's remember what happened at the end of the citric acid cycle, or what those products were. At the end of the citric acid cycle, we had a little bit of ATP that was produced, but more importantly, we had a bunch of electrons being carried by those enzymes. Specifically, the enzymes that were carrying all that energy, all those electrons, were NADH and FADH. Or FADH2, rather. So those electron carriers, those enzymes, NADH and FADH, end up within the cristae, within those folds of the mitochondria. And what they're going to do now is the enzymes end up within the cristae, but so do these hydrogen ions. Remember, the hydrogen ions are really important. So what's going to happen now are these electron carriers are going to transport their electrons from one protein to the next. So you see when this NADH right here drops off its electrons, it gets recycled to NAD, right? So it can do its job again. And FADH2 does the same thing. It drops off its electrons to one of those proteins in the transport chain. And from there, those electrons just go from one protein to the next. So this is essentially that electron transport chain. And at each point when electrons get transported from one protein to the next, energy is released. Okay, well, great. But what is that energy used for? The energy is used to, let me get a better color here. The energy is used to pump hydrogen ions across the membrane. So remember I was saying that that hydrogen ions are really important in this process, not just the electrons and their energy they represent, but those free hydrogens are super important. And so when those electrons get transported from one protein to the next, the energy that's released is used to pump those hydrogen ions across the membrane. And why is energy necessary for that? Because these hydrogen ions need to be pumped across their concentration gradient. Look how many hydrogen ions you have on one side of the membrane. You've got a lot, right? And what we know about diffusion is that molecules want to move from an area of high concentration to an area of low concentration. So these hydrogen ions really want to move back across the membrane in that direction, but they're not allowed to. And so it's kind of like trying to swim up a river or swim up climb up a waterfall. You're going against resistance, and that's kind of what these hydrogen ions are doing. So there's energy required to move those hydrogen ions across the membrane. They're swimming up river to get across that membrane. So we're setting up a concentration gradient here. We are taking those hydrogen ions that are free inside the cristae, and we're moving them across the membrane. That overall sets up this concentration gradient where you have a high number of hydrogen ions on one side of the membrane and a lower concentration of hydrogen ions on the other side of the membrane. Okay, great. So that's where the energy goes to. That energy goes to pumping hydrogen ions across the membrane against their concentration gradient, creating this difference between hydrogen ions on one side versus the other. The importance of this is when we have this gradient set up is that the second process or the second substage in oxidative phosphorylation is chemiosmosis. We've just set up this concentration gradient for chemiosmosis to occur. What's going to happen, and it only happens through this enzyme ATP synthase, is those hydrogen ions normally can't just pass directly through that membrane. Those hydrogen ions have got to pass through ATP synthase. So they can only go through ATP synthase, this enzyme, to get to the other side. And it's explained in your book that ATP synthase is like a turbine, or kind of like a turnstile going into a stadium, that as hydrogen ions pass through ATP synthase and make their way back into the cristae, the ATP synthase then kind of turns in a way, and when it does that, it creates enough energy to phosphorylate ADP. So it creates enough energy to add phosphate to ADP, and that's how we get ATP. ATP synthase is so efficient at this, ATP synthase is so good at doing this, or so efficient, that's why so much ATP is produced through chemiosmosis. So really the electron transport chain here is used to shuttle electrons from one protein to the next, just to set up that hydrogen ion concentration gradient. Then those hydrogen ions are able to move through ATP synthase, creating enough energy to phosphorylate ADP to create ATP. Okay, I know this got a little bit messy, But go through this process. Watch this part of the video a couple of times. Pause the video without my drawings on it. Go to the PowerPoint and look at the blank slide. See if you can go through the steps of oxidative phosphorylation. First, with the electron transport chain. Second, understanding what happens in chemiosmosis. Remember, the electron transport chain is where energy, just enough energy is released to pump hydrogen ions across the membrane to set up the gradient. And then those hydrogen ions pass through ATP synthase, and as they do, they create energy to phosphorylate ADP, and that's what makes these large amounts of ATP. Okay, so if we just kind of summarize cellular respiration, remember we've got glycolysis, the initial splitting of glucose, and that doesn't make a whole lot of ATP, but it does make the pyruvate, which goes into the citric acid cycle, and we've got 2 molecules of NADH. Those 2 molecules are the electron carriers, the energy carriers. Pyruvate then goes into the citric acid cycle, and what comes out at the end of the citric acid cycle are 2 molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2. Remember that it's the NADH and the FADH2 that are produced in these 2 stages that are going to go into the last stage, oxidative phosphorylation. So all of that energy gets sent to that 3rd stage, oxidative phosphorylation. And the 2 steps in oxidative phosphorylation, remember, are the electron transport chain and chemiosmosis. At the end of chemiosmosis, what we have, the products are 28 molecules of ATP, so much more ATP than is produced in glycolysis and the citric acid cycle. Remember, in each one of these steps, water is a byproduct and carbon dioxide are also byproducts. So if we were to look at the equation one more time, the overall equation, we start out with a molecule of glucose, and in the presence of oxygen, we need oxygen, right? Why do we need oxygen? Oxygen is the terminal electron receptor. Oxygen is going to accept those electrons. So the byproducts, the things that we can't really use that aren't high in energy, are going to be CO2 and water. Those things are going to be exhaled. And ultimately, ATP. And it's that ATP that we're really after, right? And then also, because cellular respiration is only about 34% efficient, meaning that it only is able to harvest about 34% of the energy in glucose, that means some of that energy is not turned into ATP, but rather it's released as heat. So heat is also a byproduct of cellular respiration. Okay? So if we look at everything all combined, you see all of the steps, where they occur, what the reactants are, and what the products are. Let's recall that glycolysis occurs in the cytoplasm, and at the end of glycolysis, you have 2 molecules of ATP produced, but more importantly, we're going to send pyruvate into the citric acid cycle and send the electrons, that energy, ultimately into that very last step. Okay, now pyruvate is 2 molecules of pyruvate. I'm going to write that here. 2 molecules of pyruvate are going to go into the citric acid cycle. The citric acid cycle is going to turn, and the citric acid cycle is going to produce a whole bunch of NADH and a whole bunch of FADH2. Again, electron carriers. That energy gets sent into oxidative phosphorylation. And through oxidative phosphorylation, both steps, the electron transport chain and chemiosmosis, out the other side, once oxidative phosphorylation is finished, you get about 28 molecules of ATP. So overall, the maximum amount of ATP that cellular respiration is able to produce for 1 molecule of glucose is somewhere around 32 ATP. You might see variations in this number a little bit depending on which video you watch or which textbook you read. So go with 32 because that's what our textbook says, even if there are variations. Sometimes you'll see a variation. You'll see that, someone might say, cellular respiration creates 30 ATP. And sometimes the reason they say that is because 2 of these ATP that were initially produced here are actually used up in getting this whole process going. So the overall amount of ATP produced per molecule of glucose at most is 32. So we'll just go with that number. Okay, so in parts 1 and 2, we spent a lot of time understanding the process of cellular respiration. And sometimes that's called aerobic cellular respiration, and aerobic just means to utilizing air, using oxygen. However, there is another way that energy can be produced through the conversion of glucose that doesn't require oxygen, and it's called anaerobic. So if we look at this prefix right here, an means without. So that essentially means without oxygen. And some organisms can carry out anaerobic respiration or anaerobic cellular respiration. And one of the ways in which they do that is called fermentation. And so fermentation is cellular respiration in the absence of oxygen, no oxygen available. So it's a way to take advantage of glycolysis. We can still split glucose and make a little bit of ATP. So there are 2 molecules of ATP produced when you split glycolysis. The thing with aerobic cellular respiration, though, is there is no citric acid cycle and there is no oxidative phosphorylation. There is no oxygen there to accept those electrons. So really, fermentation is just glycolysis, just that first stage of cellular respiration. So we produce 2 molecules of ATP per molecule of glucose, and then we reduce NAD plus to NADH. So NAD plus accepts hydrogen and its electrons. So remember that at the end of cellular respiration, aerobic cellular respiration, that we have about 32 molecules of ATP produced. Well, fermentation doesn't produce nearly that much energy. It only produces about 2 ATP. But you know what? It's energy. So, that's good. So with fermentation, though, the trick is to provide a way for the enzyme NAD to be recycled. We reduce NAD to NADH, but we've got to find some place for those electrons to go. Remember, in aerobic cellular respiration, oxygen is the terminal electron receptor. So in fermentation, now we've got NADH carrying 2 electrons. We'll just draw them in right here, carrying 2 electrons, but without oxygen, there's no place for those electrons to go. So this is where it gets a little bit tricky. So let's go ahead and let's look at back to here, right? So in lactic acid fermentation, the lactate that's produced as through that process is carried in the blood to the liver. And then the liver converts that back to pyruvate and is ultimately oxidized in the mitochondria of liver cells. You're familiar with lactic acid fermentation. Dairy industry uses bacteria to carry out lactic acid fermentation to make cheese and yogurt. So if you like cheese and yogurt, you can thank bacteria for carrying out their lactic acid fermentation. There are other types of fermentation that are carried out by other different microbes that turn, for instance, soybeans into soy sauce or cabbage into sauerkraut. This picture first. Okay, so there are two types of fermentation. The type of fermentation that is carried out totally depends on the organism that we're talking about. But let's look at both. Let's look at some of the similarities here. So each type of fermentation begins with a molecule of glucose. And glycolysis, remember, is splitting a molecule of glucose. So we are going to split a molecule of glucose right down the middle. So that is the same. And what is also the same is we've got the enzyme NAD+. We've actually got two of those. So when we split the glucose molecule in glycolysis, we're breaking bonds, releasing hydrogens, and releasing the electrons associated with those hydrogens. So NAD+ in both types of fermentation NAD+ gets reduced. It gains hydrogens and gains electrons. So look, there are those electrons, and there are two of those molecules. And so you've got two molecules of NADH hanging on to two molecules of hydrogen. And that's the same in both. So when that occurs, just enough energy is released to phosphorylate ADP. Look, we've got two free phosphates that are going to be added to ADP. Remember, that's phosphorylation. So the products are two molecules of ATP. So there's the energy that can be produced through this type of cellular respiration. The products that are produced are the same also. After glycolysis, you have two molecules of pyruvate. Okay, so that so far is the same as everything that we've learned, and it's the same between these two types of fermentation. Remember, the trick is we need to figure out where these electrons go. There's no oxygen to take those electrons from NADH, but we have to recycle NADH to NAD, the enzyme, so the process can continue, right? So this is where we start to see some differences. The difference here being that in lactic acid fermentation, over here on the left, in order to recycle the NAD, what happens is pyruvate picks up the hydrogens and the electrons from NAD and gets converted to something called lactate. So this is why we call this lactic acid fermentation. In alcohol fermentation, what happens is in order to recycle the NAD+, the electrons get released and they create bonds here, but also two molecules of carbon dioxide are released. So essentially, look over here on the left where you have a 3-carbon. In alcohol fermentation, those 3-carbon molecules, the bond is broken right there, and you get 2 free carbons. That's where you get the CO2. I'm just showing you, just using the 3 carbons over here to show you what happens in alcohol fermentation over here, right? So here, I guess I can show it to you here. 2 of these carbons get broken off from pyruvate, and that's how you get the 2 molecules of CO2. And then what you have left are 2 carbon molecules that accept those electrons, and those 2 carbon molecules are ethanol, the alcohol that you drink. So this is why this is called alcohol fermentation. CO2, as a byproduct of alcohol fermentation, is the reason why beer or kombucha or many other fermented alcohol drinks are fizzy. This is where they get their carbonation from, this process of alcohol fermentation. Okay, so one last thing I want to cover with you then is let's look at the different foods we eat, the major macromolecules, and let's see how they get utilized to produce energy. We've gone through extensively how carbohydrates get used as energy. Ultimately, the carbohydrates that you eat get broken down into monosaccharides like glucose, and we followed that whole process, right? We know exactly what happens to glucose and how much energy is produced from glucose, but we also consume fats, and fats can be used as energy. Even though your body would really like to use glucose first, if there's no glucose present, if you've ever been on like a low carbohydrate diet, if there's no glucose present, you don't just die. You can also metabolize fats. Fats can be used to create energy. Proteins can be used to create energy. Although metabolizing fats and proteins doesn't produce nearly as much ATP as does metabolizing carbohydrates. So if we look at fats, if we break down fats into their monomers, glycerol and fatty acids, the glycerol from fat can go right into glycolysis, and the fatty acids enter the citric acid cycle. So they can be broken down to produce ATP. And in some cases, depends on the type of amino acid, but proteins can be broken down into their monomers, amino acids. Some amino acids go directly into the citric acid cycle. Some amino acids get converted to pyruvate and go directly into, not directly, indirectly into the citric acid cycle. And then others go way back here, get converted into pyruvate, and then get converted into acetyl coenzyme A. So those things that we consume or other organisms consume, the major macromolecules, carbohydrates, fats, and proteins, all of those can be used to produce ATP, but carbohydrates are the ones that produce the most ATP. Okay, the last couple of slides here are just things that you should now be able to understand or do. So I'm not going to read these to you. I'll just click through them slowly so you have them to, you can pause your video and look at these. These same slides are on the PowerPoints too they have available to you. Okay, that wraps up cellular respiration. Next time we will learn about photosynthesis. Remember, photosynthesis is the way that, or one of the ways that plants produce energy. Okay, that's all I have. See you next time.